Hello and welcome everyone to this video podcast for the lab of Biology 211. In this lab we're going to start working on our third module. And in this module we're going to work with yeast. And more specifically we're going to see how researchers use yeast to identify specific genes in specific pathways. For instance, in this lab we're going to identify mutants that have a decreased ability to grow in the presence of stresses. Now remember, yeast are single-celled, much as you see over here on the left. However, when we work with them in the lab, we don't usually work with them at this scale. Rather, we work with them as they grow in colonies on plates, as seen on the right. Down here, we can see how we have wild-type yeast, and then we have mutant yeast. And in this particular case, the mutants can grow on the control plate with no stress on them. But as you add stress to these plates, you can see they have a decreased ability to grow. That's similar to what we're going to look at today in our genetic screens. Up here you can see I made all caps screens, because that's what we're doing today. But as you read in your manual, you learned that there are also genetic selections. But we're going to be doing screens. Okay, I want to kind of break down this week for you in the different sections of it. It's going to be a busy week, and I really want to encourage you not to put this all off towards the end of the week. I want to encourage you to break it down into 3 separate units, much as you would if you were attending a lab. First, let's talk about Lab 3.1. The rest of this PowerPoint will be devoted to 3.1, but more specifically, we're going to talk about how we're going to use yeast to construct a genetic screen. And then we're going to spend some time looking at some yeast bioinformatics, and then you should start working on your poster. Again, please don't put your poster off until the end of the week. Rather, work on it a little bit at a time. Then Lab 3.2, which will be covered in the next PowerPoint, we'll spend some time looking at how we're going to analyze the results from your yeast genetic screen. And then you'll have the rest of the time to work on your poster and hopefully complete it. And then the last part of this week will be Lab 4.1. And that's a lab where we're going to start working with DROSOPHILA, at least in a virtual sense. So we'll talk about DROSOPHILA mutants. And then there is this program called STAR Genetics. And we're going to use that for some virtual crosses. And in fact, this is what we do in the regular face-to-face labs as well. Just to kind of give you a framework for this week, what's due, since I do think this is a pretty busy week in lab. The lab quiz for this week will be due Friday. It has 15 questions, 30 points. Usually this is due Thursday, but I'm kind of extending some things out to give you each that freedom to work it into your schedule. There are 2 worksheets this week. I could have made them 1, but I decided to break them up into 2. And the first one is based upon Module 3, working with just the yeast. And that one is going to be due Friday at 11:59 p.m. And then on the bottom, I'm going to skip this third point here for a moment. Also on Friday, your final yeast poster is due. And you'll upload that to Blackboard. All right, so now the second set of worksheets for Drosophila, that will be due on Saturday at 11:59 p.m. I'm giving you an extra day to work on that compared to the other assignments because I really want to make sure you spend some time on this because I think it's really going to help you understand the material that we're covering in lecture. So these worksheets really dive pretty deep into Mendelian genetics, and that's what we're covering this week in lecture. The lecture exam over the Mendelian genetics will be that Monday. And so this will give you a lot of practice so that you're prepared for the exam. At least that's my plan. So now let's look here. I just want you to kind of keep a note of this. This is different than what's in your lab syllabus. It's not terribly different, but some of the point distributions have been changed. Just a little bit, not a lot. And I've also added some more clarifications with some things. You'll notice that our week 6 here, that's when the poster evaluations are due. So I put that off until week 6. Now we also have something else to do in week 6, and I'll provide some more information on that later. But my idea is that it will not be too difficult to do. All right, let's spend a little bit of time talking about genetic screens and genetic selections. Let's begin with a genetic selection. In this example here, we have a plate of yeast colonies. Then some sort of genetic selection is applied. For instance, maybe, as the example over here says, maybe it's a poison that's added. Now with a selection, only the colonies that survive here are the ones that we're interested in. These 3 mutants here have something in them, some kind of mutation, some kind of rare mutation in them that allows them to survive. All of these other ones that didn't survive, they didn't have the mutation that we're interested in. They didn't have the mutation in them that would allow them to survive. So a genetic selection is very useful because it quickly identifies the mutants that you're interested in studying. However, genetic screens are commonly done because they help identify some of the mutants that have less clear phenotypes. A genetic screen is going to tease that apart a little bit more. So again, you start off with a similar plate here of yeast colonies. You apply a genetic screen to it. Now, it could be a poison like we had in the first one, but the key difference is that in a genetic screen, many of your cell colonies will survive. In fact, most will. And because most of them survive, it's not quite as obvious which ones you're going to be interested in. So they have to be sorted out. So in a genetic screen, you might say, look at this one's huge, this one's bigger than all the others. So maybe there's something interesting in that mutant. Maybe I didn't have one of these in the plate here, but what if you had one that was really small, a colony that was really small? That also might be something you would want to sort out. There might be a difference in shape, a difference in color. All of these could be phenotypes that you could use to sort out mutants of interest. So more complicated than a selection, but often yields a greater variety of mutants. We're going to talk about how we would conduct a genetic screen using yeast. As we begin to think about this, remember that the yeast genome is relatively small for a eukaryote. There are only 6,000 genes, approximately 6,000 genes. We have a collection of about 4,500 yeast mutants, and they're stored on these 96 well plates that we keep at minus 80 degrees Celsius. We often call a collection of mutants like this, call it a library. We'll see a picture of this plate in just a moment, but these plates, as I say up here, have 96 specific wells. Each well contains a different mutant. Now, real quick here, you might have wondered, if there are 6,000 genes in yeast, why do we only have 4,500 mutants? And the reason for that is that somewhere between 1,000 and 1,500 genes in yeast are essential, meaning if we mutated them, the yeast cell would not survive. And if they don't survive, we can't hardly study them. And they can't form a colony to store in a plate. So there are some genes that are just a little bit more difficult to get at, and that would be these essential genes. Non-essential genes are in these plates. Now, each team will only screen a portion of these yeast mutant library plates. And so that means out of the approximately 6,000 genes in yeast, we're only going to look at 94 of those mutants. In the screen, you're going to look at data to help you identify yeast mutants that grow slower or faster or not at all in the presence of an oxidative stress, like hydrogen peroxide. Now, even though it's a 96 well plate, there's only going to be 94 mutants in them because two of the other wells contain our controls. So one of those wells will contain wild-type yeast, and the other well will contain no yeast. All right, so let's talk a little bit more about this. Each well of the 96 well plate contains a culture of yeast. As I said on the last slide, most of these wells will contain a culture of yeast that has one gene mutated in them. This is an important thing to remember, and that is how these mutants are categorized. So we have our columns here, of course, and then we have rows here. And so it's A1, A2, A3, A4, all the way down to A12, and then same thing with B, B1 through 12, C1 through 12, and all the way down here to H1 through 12. This is important because we'll see later on that if we know there's a mutant of interest, say at H6 here, we can use this identifier of H6 to know exactly which gene is mutated. It's a very powerful but rather simple way to categorize the mutants. Now, since the lab is online for this summer, everyone's going to work with plate 116, so we can show you the data from this one set of experiments. As I said before, we have two controls. Wild type cells are in well H3 of every plate. It's H3, and so we would expect growth from this well here. So we call that the positive control. Now no cells are in B4, so that's our negative control. Now depending on which plate is used, the negative control changes. So this is plate 116, and so that means it's the 16th well. So you have our first 12 rows here, then 13, 14, 15, 16. So if you were working with plate 124, you would look for the 24th well. So 12, and that would be this one. So the last two digits here represent the well in numerical order going from left to right. But for 116, we know it will be here at B4. So unfortunately, you can't do this with our online lab, and there's not a good simulation for it. But I'd be happy to show you how we do this anytime when we're allowed to get back on campus. But we have this metal tool here that we call the 96-pin replicator there are stainless steel pins all along here, going from the bottom of the base straight down. And they are spaced in such a way that you can hold this up right above this plate here. And this plate here would have all of our yeast cultures in it. And then you place each of these wells simultaneously in each of these wells at the exact same time. And then you lift the 96 pin replicator straight up and move it over to the YPD plate, or the YPD plate with the stress on it. And you just gently place that onto the YPD plate and pull up the 96 pin replicator. And in doing so, in about 10 seconds, you've inoculated this plate with 96 cultures of yeast all at the same time. And actually I should have said 95 cultures, because one of those wells should be blank with no cells in it. Just real quick, I just casually threw out YPD, but I should have defined what that was. This is just the standard growth media that yeast grow on. That's all the nutrients they'd want. It's like a yeast smorgasbord buffet. They love this stuff. Now you add a stress to it, they don't like it as much. Now each team will examine the results from 3 different plates. YPD, all yeast should grow on this one, so you should have 95 colonies growing on the YPD plate. Sometimes you will get a few cultures that just don't grow. Then you're going to have your stress condition on the hydrogen peroxide. Now in this, we would expect most of the cultures to grow. However, if our screen is set up right, we would predict we would have a few of these cultures that would have an altered growth pattern on the hydrogen peroxide. And that's part of the screening process. You'll have to determine which of those cultures are of interest. We also do a YPG. This is kind of a backup in case the hydrogen peroxide plate doesn't work. So after you look at the hydrogen peroxide plates, if you don't find anything on there that looks interesting, then you can look at the YPG plate. I can tell you though, we picked plate 116 because we know that there is at least one mutant on there that has an altered growth pattern. Now, on Blackboard, you'll see artificial data from plate 116. You will want to download those images so that you can examine them and analyze the growth patterns. So in your experiment, there are several controls. There are positive controls on each plate. So well H3 has wild type yeast on each plate. We would expect growth on that plate, in that well. The negative control. Each plate has one well that identifies its number that has no yeast in it. And this will be, for plate 116, B4. And then we also think of the YPD plate as serving as a control. We would expect all the mutants to grow on YPD plates. Alright, so I hinted at why we also do use a YPG plate in this experiment. And if this were a research experiment going on in my lab, we would just keep going through all of those plates until we screened all 4,500 mutants. And at that point, I'm confident we would have gotten a mutant. In fact, we've done that screen of all of them, and we know which ones will or will not grow on certain kinds of stress. But we don't have that kind of time in a semester-long lab. So we predict you should find something on the hydrogen peroxide plates. But if that doesn't work, we know you'll find something that doesn't grow on the YPG plates. So if you don't see something growing on the YPG plates, why is it not growing on YPG? And I should back up here a moment as we think about this. The reason we call it YPG, the G stands for glycerol. The D in YPD stands for dextrose. And dextrose is just like glucose. So this is a very standard sugar. Glycerol, however, is not. Yeast can only metabolize glycerol if that yeast can undergo respiration. If it cannot undergo respiration, it will not grow on YPG. All respiratory deficient mutants will grow poorly on YPG. And when we say poorly, we mean not at all. If you can't respire, you cannot use glycerol. These mutations are somewhat common. Of the mutants that you'll be looking at, some of them will be respiratory deficient. Now, as I said before, we're going to be working with artificial data, but it's real data just from a previous semester. There are 3 files on Blackboard for plate 116. Some of the data will contain growth on YPD, some will contain YPD plus hydrogen peroxide for our oxidative stress, and then some will contain YPG. And we have it set up for days 1, 2, 3, 4, 5, 6, and 7 for each of these. So you'll have to go through each of these to see which mutants are growing well on YPD, but less well on hydrogen peroxide, or less well on YPG. And we'll go over that a little bit more for lab 3.2. And just to remind you, here are our controls, H3 for our wild type cells, and B4 for the no cells. On Blackboard, you will see these worksheet data collection sheets, and let me show you what they look like real quick. Now as you're looking at all of these plates for each of the days, it might get a little confusing. And so I've included some data collection spreadsheets for you on Blackboard. You don't have to use these and you don't need to turn them in, but it might make life a little easier as you're evaluating the data. However, you may just want to look at these images just like this and go straight from that. If that's what you want to do, that's fine. However, some of you are going to want to record the data from these plates and put them on these data collection sheets. I tend to think that might be helpful, but you don't have to do it. You will have data collection sheets for YPD plates, YPG, and YPD plus hydrogen peroxide. You can record the data any way you want. However, a convenient way to do it would be minus for no growth, plus for just a little bit of growth, 3 pluses for really crazy amounts of growth, and then somewhere in between patterns of growth, you would give it 2 pluses. It's really up to you how you want to do that though. So as you're analyzing the results, it might be useful to see an example plate here. So this is the YPD plate, and it's not a YPD plate from plate 116, and you know that because our negative control is here. And so we know this has to have come from which plate? Let's see, so we have 12, 24, 36, 47. So we know this came from plate 147. Our stress condition plate also has no growth there. And so that's an effective negative control. It doesn't grow on either plate. Our positive control in this experiment, this was set up a little different than the one you're doing. The positive control in this one was in H12 as shown here. All right, so we can see that our controls are working, but now let's look at this red square identified here. If you see something like this is of immediate interest. You can see that this colony here is growing on YPD, but it's not growing when you add a stress to it. That tells you immediately that the gene that is mutated in this colony here isn't necessary to grow on YPD plates, but it's necessary to grow when you add a stress. So this gene is important in surviving a particular stress, and so it is of immediate interest. Now the question becomes, how do we know what gene is mutated here? And we want to think about yeast bioinformatics to address that question. So let's talk a little bit about bioinformatics here. Every model organism that has their genome sequenced, at least all the ones I know of, has some sort of internet-based database where all that sequencing information is stored in a usable way. So anybody can access it and quickly look up genes and gene information. Now you're going to spend a little bit of time going to this website for yeast, yeastgenome.org. That is the bioinformatics website that we go to study yeast genes. You're going to complete worksheet 3.1, part 3, to become more familiar with this website, because you're going to use this website to analyze your data in your screen. So remember on the last slide, A2 we knew was of interest. And so how are we going to use that here? Well, let's just take a little example here. On Blackboard, you will have this Excel file. And on this Excel file, you will have every single gene that is on plate 116, only showing 14 of them here. But for instance, let's say we were interested in B2, similar to what we saw on the previous slide. B2 had a growth condition that we were interested in. And so you come over here and you can see that the ORF name, or the gene name, is this, YLR181C. Not a very fun name by itself, but that's what it's called. We take that and we plug that into the yeast genome database. So let's go ahead and do that real quick, just as an example. Okay, so here's our Saccharomyces genome database, yeastgenome.org, and here's a search feature up here in the right corner. And so we're going to put that ORF name here, YLR181C, and we're going to search for it. And then what we'll see is a lot of data that comes up. We can see that its standard name is VTA1. That usually tells you a little bit more about its functioning. It tells you a little bit about it, gives you a description of what this gene does. It'll tell you different things about where it's located in the genome, what genes are next to it, so you can see who its neighbors and who its friends are. It also gives you some information about the protein. When this gene makes a protein, it has 330 amino acids in it, its molecular weight, all this other data with it. Down here it'll tell you that the null is just a way of saying when the gene is completely absent, so a complete mutation. What does that do to the yeast? It tells you that the protein peptide distribution is abnormal and there is a defect in vacuolar transport. And through other screens we can say it's already been identified as having a poor growth in anaerobic conditions, cell size is increased in these conditions, and so we can see a lot of different things like this. Toxin resistance is decreased. So that's pretty consistent if we know that this gene mutated in our screen doesn't grow well under stress, it's already been shown that there is a toxin resistance. That kind of helps validate our screen. And I won't go through everything here, but there's a lot of information here just on that one gene. And then there's also a lot of references here. So this would be a good place to find your references when you're looking for references for your poster. But when you complete that worksheet, you're going to be asked to plug in different information here and pull out various pieces of data. All right. And then what you should do for Lab 3.1 is to begin thinking about your research poster. Posters are a common way that scientists communicate their research results. In fact, most of the meetings I go to, it's the poster session where I get the most relevant and up-to-date research reports. It's not always the talks. The posters are an essential part of understanding that cutting-edge research. A poster, just like a research paper, will tell a scientific story. It'll also have the same sections that a lab report will have. Though as you will see, those sections are abbreviated. To understand the poster a little bit better, I'd recommend going to Chapter 10 in the Macmillan Writing Manual and the poster rubric. Now I've added a revised poster rubric on Blackboard because we had to adjust it a little bit because there's not going to be a presentation like we have typically done. So look at the revised rubric on Blackboard. Some key things to keep in mind when you're thinking about a poster: it should be readable from a distance. So a lab report might be written in a 10 or 12 point font. The font for a poster, depending on the section of the poster, might be anywhere from 24 point font all the way up to a 48 or 72 point font. Choose professional colors of background and font. So it's okay to have colors, but you want to make sure that it doesn't detract from the science of the poster. And you also want to make sure it's readable. For instance, some colors have a very good contrast. For instance, black and white has a good contrast, as you can see on this slide. Also, green and white, as you see up here in the title. That's easy to see. Those two colors don't blend with each other. Some colors to avoid is to have, say, a blue poster with yellow font. That's really hard to read. Use consistent font type throughout your poster with the correct sized fonts. You don't want to have one part of your poster in Times font and another part in Arial. You want it to be consistent. As I said, a poster tells a story, so there should be a logical flow. As somebody's reading your poster, they should almost be able to anticipate what the next part of your poster is going to be. In a lab report, you're taught to write in paragraphs, in well-structured paragraphs. However, poster presentations are a little different. Most people don't have the time to spend more than, say, a few minutes at your poster, so they're not going to have time to read a long paragraph. In fact, if it is a long paragraph, there is a possibility that people just won't read it. And that's why bullet points are important. The bullet points are typically full sentences, but it gets straight to the main point of that figure or the main point of the introduction or whatever section you happen to be on. As with anything you write, avoid spelling and grammar errors. Use your space well. You don't want to have a big blank area in one part of your poster and a whole bunch of words and picture crammed into another area. It should be well balanced and easy to look at. Starting your research poster. Work together with your lab team. You should be doing this with all of your things, but in particular this one because you're going to turn in one team poster. You're not going to turn in 4 separate posters or 3 separate posters depending on the size of your team. Just one poster. Often when we have team projects like this, some students are concerned that not everybody is contributing equally. There will be an opportunity for you to evaluate your team members so that everyone participates in an equal way. I have a poster template on Blackboard. Use that to start with. In fact, let's just go ahead and look at that poster template. So here's the template found on Blackboard. One thing nice about this is if you use it, all the fonts are going to be in the correct size. So you know you won't lose points for that. It shows you all the sections you need to have. Again, the font type or size for the headings is one size and that for the body of it is another size. It's already set up for you. The figures are also given to you in a way that makes it easier to fill in the rest. Obviously, your figure is not going to be a blue box. You're going to have to actually put in the figure that you want from your Yeast screen in each of these. But this should make it a lot easier to complete the poster. Now you'll see here I have these margins set. These are so that you don't go over these spaces. You always want to make sure you have a little bit of margin on either side because it's not very pleasing to look at with everything squished all the way to the side here. And plus then when it prints, sometimes it can be blurry. So we always try to leave a bit of a margin here. Before you turn in your poster though, delete these. Again, read the lab manual and the revised poster rubric to help you understand the different parts of the research poster. It has to include a title, introduction, methods, results, 2 figures in one table, conclusions, and references. Points that were originally designated for the presentation and time categories for the poster rubric have been redistributed elsewhere. So that's why I have the revised rubric on Blackboard. Okay, so remember, you're working in your teams on this. Take advantage of the Microsoft Teams space online to work together with. I'd recommend putting the poster right there in your files, and that way everybody can see it and you can work on it in real time with each other. So looking ahead to the next lab, Lab 3.2, read the following sections in the lab manual and Macmillan. Lab 3.2 for data analysis and poster presentations in Chapter 10. Understand how to interpret yeast screens if given sample data, and then you're going to finish your poster. At least get most of it done by then, because if you can get your poster done, say by Wednesday of this week, that'll be a huge thing off your plate, and you'll be able to relax a little bit more this weekend. All right, that's all I have for this podcast. If you have any questions at all, make sure you contact myself or your TA, and we'll be happy to answer any questions you might have.